topic, heredity part 2. So here in this lesson I have cut it short into a very small portion because as heredity is a new part and it is quite difficult to understand. So when you are aware with the stages of Mendel's experiment and monohybrid cross, then only we can move on to dihybrid cross. So today at the end of this lesson we will know that what are the stages of Mendel's experiment and what is monohybrid cross. Now I think in the previous slide I have discussed about the 7 pairs of contrasting characters of a pea plant. So I think you do remember that. If you don't remember that, please go through that video again before starting to read this video, or else it will be difficult for you to understand these experiments. So let us start with the experiment. Here we are starting with stages of Mendel's experiment. So this is stage 1. So what did he do first? Now Mendel first selected 7 pairs of contrasting characters as I have told in the previous video. Now here what did he do? He took pea plants and self-pollinated them for several generations. So what does this self-pollination means? That is, pollination is happening within the same plant. So within 2 flowers of the same plant, pollination is happening. So in this, plants were self-pollinated for several generations. So there, what he observed? He observed that seeds from the tall plants produce only tall plants, and the plants with purple flowers which were self-pollinated were all producing purple flowers only. So in these 2 self-pollinated cases, there is no contrast characters which are appearing in this after the plants grow. So what we are observing, that is, thus he obtained pure line. So obtained pure lines means plants which are breeding true for selected characters. They produce the same kinds of plants. If they were tall, the plants which were produced were tall, and if they were having purple flowers, the plants after the several generations also, as they were self-pollinated, they were having the purple flowers only. So this was the stage 1 of Mendel's experiment, and he did self-pollination for several generations. After what he saw, tall plants after self-pollination produce tall plants, and purple flower plants produce purple flower plants. So is it clear for everyone? So now we will move to stage 2. Now here what he did, he crossed 2 plants having different contrasting expression of a single trait. So he conducted reciprocal crosses also. Reciprocal cross means crossing with contrasting features. Now example, a true breeding tall stem plant was crossed with a short stem variety or a dwarf plant. So there was reciprocal cross done. So a tall stem plant was crossed with a dwarf plant or a short stem variety. So this is what is happening? Here a cross between 2 parents or 2 separate plants is happening, representing contrasting forms of a single trait. Means one plant was tall, another plant was short. So they were crossed. So 2 parents representing contrasting forms of a single trait. So what is the trait here? Their height. Both tall and dwarf are the single trait. So only one trait or one character has been taken to do the crosses, or the feature is called the monohybrid cross. Now here the offspring which is produced is called the hybrid. Now when 2 traits or 2 characters are taken to do this cross, we will say that it is a dihybrid cross. And when 3 or 4 traits are taken, it will be called trihybrid and tetrahybrid crosses, and so on. So is it clear for everyone? He took now 1 pair of contrasting characters between 2 different plants. It was not a self-pollination. Here it is cross-pollination. So between 2 different parents, and Between one contrasting character only it took place, and it is called a monohybrid cross. The product form is called a hybrid. Now if we are taking two certain characters, they are called dihybrid cross, and 3 characters, we know 3 refers to tri and 4 refers to tetra. So tri-hybrid crosses and tetra-hybrid crosses, and simultaneously it goes on. So let us see what is there in the next slide. Here comes the main step of cross-pollination. So what is happening in these two flowers? Now let us see that. Now first what he did, he performed the experiment by transferring the pollen grains from the anther of the purple flower to the stigma of the white flower. So this is the purple flower. Pollen grains are transferred from the anther of the this purple flower to the stigma of the short plant. The white flower plant. Now what was done here? As it is a cross pollination happening, so we should prevent the self pollination. So to prevent the self pollination in this step, what was done? Now Mendel removed all the stamens of this white flower plant. So he removed all the stamens of this white flower plant so that there should not be any self pollination. Only the cross pollination should happen. So what he is doing? He is cutting off the pollen, is cut off and is removed, and the pollen of the white flower is taken and transferred to the pupil of the white flower, this purple flower. So pollen of the white flower is taken and it is transferred to the purple flower. Now the fertilized pistil gets matured and they become this pea pod. When this pea pod are fully matured and they are grown, what they are being done, they are ready to be planted. So when they are planted, what is happening there? Now as a result of cross pollination, we are seeing that in F1 generation, all plants are bearing purple flowers. So though the self pollination was prevented by cutting out all the stamens from the white flower plant. So here you can also contrast it with a tall plant and a dwarf plant. So in case of this purple flower, you can put a tall plant, and in case of this, you can put the dwarf plant. So any one contrasting character should be taken, and to prevent the self-pollination, the stamens of any one of the plants should be removed. So here we have done with the white flower plant, and the pollens from here are taken and transferred to the pupil of pistil of the purple flower. So this pods, mature pods were sown, and all the plants whatever grown in the F1 generation were all with purple flowers. So what was observed? The observation of this cross pollination was in F1 generation or first filial generation, the plants produced only purple flowers. So let us see the next step. Now what was the observation of the previous monohybrid cross? So the observation was all plants which grew from those seeds were tall plants. The plants in this generation were called F1 generation or first filial generation. So this is called the F1 generation or the first filial generation. And all the plants in this generation were either if we are taking the tall or dwarf plant, they will be tall plant because dwarf is a recessive trait. And if you are taking the purple flower and the white flower, as in the diagram we have seen the purple flower, and white flower, we will say that all the plants growing in that generation were having purple flowers. So all were having purple flowers and all are called F1 generation. So a first filial generation. Now here in stage 3, what did Mendel do? Mendel further planted the F1 seeds and flowers and were again allowed to self-pollinate to produce F2 generation plants. So in F2 generation, what happened? F2 generation, when the plants were produced, we see that few plants were tall and few plants were short. If we are taking the contrasting characters tall and short. Again, if we are taking characters like white and purple, then we will see that among all the flowers, few are purple and few are white. So why is it like that? So he saw that the phenotypic ratio was 3:1, like 3 will be tall and 1 will be dwarf, and 3 will be purple flowers and 1 will be white flowers. So this is the phenotypic ratio, and this generation is called the second filial generation. So previously it was the first filial generation. So is it clear? This is the F2 generation or the second filial generation. So he told that the tall plant, what is tall, is coming in more in the ratio. It is the dominant trait, and the short plant is the recessive trait. Whereas in the purple and the white, among the purple and the white flower, the purple one will be the dominant trait and the white flower will be the recessive trait. So here we can see the genotypic ratio will be 1:2:1. That I'll show you in the further slides ahead. So let us move on to the next one, and please go through your textbooks. If you are not getting the slides clear, go through it again and again. Ask me your queries and doubts, and please do always do subscribe to my channel, and don't forget to subscribe to it. Now here let us see the diagrammatic representation of what is happening in monohybrid cross step 1. So what are these? These are gametes. Okay, this T, these are dominant in nature, so it is written in capital T, and this small t, these are recessive characters. So these two are also gametes and these two are also gametes. So these two are parents. So this one and this one are parents. So cross pollination is done between these two parents. So what all we are getting in this F1 generation? We are getting all plants are tall. So this is the dominant trait. This is the tall plant, or the plant having the tall stem, and this is the plant having the short stem. Or we can say that here this is a recessive trait, or it is a dwarf plant. So after crossing it in the in one generation, we are getting that in the F1 progeny or F1 generation, we get all tall plants here. So for us it is clear. So is it clear, everyone? I think it should be clear. We can see there is a cross pollination. This capital T and small t, TT, capital T, and this small t is TT. Here also capital T is crossing with this small t. Here also capital T, small t, and here also this capital T, small t. So we are getting all the tall plants in this F1 generation, because T is the dominant trait and this T is the recessive trait which is being masked by the effect of the dominant trait. So we are not seeing any dwarf plant in this F1 generation. So let us see what is happening after self-pollination of this plants happens and what is happening in the F2 generation. Now here see, this is the parental generation. These two are parents. These are gametes and they are producing after mating. They are producing all the tall plants. Now here in the F2 generation, what is happening? This here we can see after these two plants are being pollinated, we are getting 3 tall plants and 1 dwarf plant. Now phenotypically it is 3:1, but genotypically is 1 fully dominant, 2 are hybrid, like they are having 1 dominant trait and 1 recessive character in them. So these 2 are genotypically 1:2, 2:1. So one is fully dominant, having full dominant characters. One is having fully recessive characters or one is fully dwarf plant. Other two being visibly tall, but they are having one recessive character also, which is the small t over here. So it is having a phenotype of 3:1 and a genotype of 1:2:1. So F1 generation all are tall. F2 generation or the second filial generation, it is 1:2:1 is the genotype and phenotype is 3:1. So now let us all wind up. So we have today studied with the monohybrid cross. So we have studied the F1 generation and both the F2 generation. So I think you all are clear with these two topics, and please be clear with all the steps what Mendel did. Go through your textbook which you refer. Go to my slide again and again so that you don't have any doubt. Whatever I tried to make you understand. And please go through the previous lessons of heredity also. Or if you are not understanding the previous episodes or the previous videos, please do write and share in my comment box. And please do subscribe to my channel. And don't forget to share it for your friends and others' benefit. Thank you.